All right, so for this next video, we're going to go a little bit faster, and we're going to now talk about how enzymes are controlled, right? How are they regulated? Because we don't want a runaway train of enzymes just speeding up chemical reactions everywhere all the time. So we're going to talk about how we regulate enzyme function, and then how we can predict kind of the outcome of changes when we see changes in temperature, substrate concentration, enzyme concentration, pH, and covalent modifications. We'll also talk about some other things that can assist with the enzyme itself and its job, like cofactors, coenzymes, apoenzymes, and holoenzymes, and then talk about some examples of those. We'll also look at the concept of end product inhibition, since this is something that we will see multiple times, and then talk about the concept of inborn errors of metabolism and look at some examples. So what are some things that can affect an enzyme's ability to function? And when we say ability to function, I mean basically the enzyme's ability to convert the substrate to the product and what rate, right? So is it going to go faster? Is it going to slow down? Is it going to stop? Right? That's what we're looking for when we're looking at the things that can affect an enzyme. So what are some things that an enzyme can be influenced by, just like a protein? So temperature will impact an enzyme's ability. The pH, concentration of cofactors and coenzymes. So if we have a coenzyme we require, and that is going to be, and not having a high enough amount of that's going to impact our ability to go through the reaction. So then that would slow us down. The concentration of the enzyme itself, right? The concentration of the substrate, the covalent modification of the enzyme, and then different inhibitory effects, right? So some that can stimulate an enzyme, that can turn it on, or that can inhibit an enzyme, turn it off. So let's talk about temperature's effect. Temperature usually will increase the rate of a reaction. So the higher the temperature, the faster the reaction will go, basically until we get a little bit above body temperature. At that point, the protein, the enzyme, starts to lose its structure. So if we look at enzyme activity, we see this is all in Celsius. So here's where we would get to, body temperature, right over in here. And we see that lower temperatures, lower activity, but there's a peak, right? At a certain point, it can no longer function because the heat is too great, and it's going to basically change the shape of the enzyme. And if we change the shape, especially if we lose the shape, we lose the function. We call that process of losing its 3D structure becoming denatured, denaturation. So here's our normal protein down here. We see it has like all these alpha helices, and it has a very specific kind of order to it. But if we heat this protein up too much, right, it will lose this shape, it'll denature, and now we just have this long ribbon, right? All of the nice three-dimensional structures are gone. And so this normal protein, when it goes through and becomes denatured, it no longer has those active sites built. It no longer has the ability to do its job. We can see this process in real life, right? So if you were to cook an egg, right? You've changed the structure, and that has changed the physical properties of that protein. We're also going to be impacted by pH. Now, enzymes usually have a very narrow pH range called the pH optimum, and it really depends on which enzyme you're looking at in terms of what's the optimal pH for that enzyme. It just depends. If the pH changes too much, right, then we can denature, and then same thing like with temperature, we can no longer do our job. Optimum pH, again, varies from enzyme to enzyme depending on where we find the enzyme. So let's look at this chart here. Over on the right, that shows a couple different types of enzymes, and we'll talk about where we find these and whatnot, and why their pH range is where it is. So we have pepsin, we have salivary amylase, and we have trypsin. I'm going to start on the blue end on the right here, and I'll work my way over. But trypsin here in blue, this is going to be found in the small intestine. Now the small intestine is going to be slightly basic. And so we see that its optimal activity is at the peak, and that's going to be at a slightly basic pH. Whereas salivary amylase is neutral, right? It's just your saliva. It's pretty neutral, very water-based. And so that one is going to peak, right, at something neutral. But then we look all the way over here at pepsin. Pepsin is found in your stomach. It's part of your stomach acid, the gastric juice. So already, just based on me saying that it's found in your stomach acid, you're probably already thinking, oh, well, it's in an acidic environment. And that's why this one, pepsin, is going to peak at something that is just over 2. Very different pHs, but because they're found in very different environments within the body. Enzyme concentration will also have an impact. So as you increase the concentration of enzymes, so you have more enzymes, then the rate of reaction will go up as well. And that makes sense because the more enzyme molecules you have, the more the substrate can come into contact with the enzyme and work, right? If you have, let's say you need to make 100 bracelets and you have 2 people working, right? Each person is going to have to work on 50 bracelets to get to that 100. But if you have 50 people working, each person only needs to do 2 bracelets. So you think about like which group is going to get done faster making the 100 bracelets? The group of 2 or the group of 50? It's going to be the group of 50, right? So enzyme concentration, the more enzymes you have in that space working, the faster that reaction will happen. Same thing with the substrate concentration. As the substrate concentration goes up, we will see that the rate of reaction will go up too, until we get to a point where the enzymes are saturated, meaning more substrate will not increase the speed of the enzyme working. The enzyme has a peak performance, right? It can only work so fast. And so if you have a large amount of substrate, the enzyme has all that it needs to work on, but it can't go past its own speed, right? But if there's very little substrate, then the enzymes are kind of sitting around going, okay, well, I gotta wait until I get more substrate to be able to do this, right? So this leads to what we call a plateau, right? So we see the saturation point. We've increased the substrate concentration this way, but at a certain point, the rate of reaction no longer changes. We don't get any faster because the enzymes are limited in how fast they can work. So in order to keep this rate of reaction increasing with an increased concentrations of substrate, we would have to increase the concentration of the enzyme too. Covalent changes to enzymes also can regulate their activity. So like when we look at the stomach, right? We look at some of the enzymes in the digestive system. We look at some of the enzymes just all throughout. They usually have an inactive form and then get activated. So for example, the pepsin we talked about earlier, that's actually the active form. It's found in an inactivated form called pepsinogen, right? And you see that zymogen, you'll see like an ogen part to the name. Fibrinogen goes to fibrin, right? Pepsinogen to pepsin. So if you see that, then you know, okay, we're talking about the inactivated form, and we're going to have to go through an activation process in order to do what we need to do. So we have that, where we can have inactivated forms that have to be activated by a proteolysis, or the hydrolysis of a protein. We can control it through turnover. Basically, the enzymes, when they're not needed, get degraded, right? Remember that ubiquitination process, right, where ubiquitin was used to mark a protein for destruction for a proteasome. So enzymes can go through the same thing, right? The enzymes are working, we don't need the products anymore from the enzyme, we just simply break the enzymes down. And then some of them require additional enzymes to phosphorylate or dephosphorylate them. So using like a phosphatase or a kinase, right, to turn on and off. Usually if you add the phosphate group, right, a kinase usually activates it. removal of the phosphate group usually inactivates it. That's not always 100% of the time, 100% of the time the case, but generally speaking, you can use that that kind of these two little bullet points, kind of keep that in your head, right? So there's a lot of things we can do, right? We literally have inactive versus active forms. We can mark enzymes for destruction, or we can use things like a phosphate group to turn them on or off. Sometimes the relative concentration of the substrate in a product can also be something used to regulate things. So enzymes, again, can drive reactions sometimes in both directions. One of the good examples is like how we use bicarbonate as a buffer, and we can go back and forth between that bicarbonate and like carbon dioxide and whatnot using an enzyme called carbonic anhydrase. Carbonic anhydrase can have this reaction go in either direction. And so this is to try to keep us kind of like in some sort of balance. If one side gets really high, then we can reverse the reaction. If the other side gets higher, then we go back, right? And so we can go back and forth between these two, right, forward and backwards, so that neither side gets really, really high. Some things that can help out with enzymes and the regulation of them are using things like cofactors and coenzymes. Cofactors are usually some sort of a metallic ion, so something like manganese, magnesium, zinc, calcium, right? They're going to help the substrate bind. Coenzymes are going to be organic things like NAD or FAD. These are going to be, usually we think something that's formed from a vitamin, and that's going to help transport small things, small molecules. And then we have these systems. So basically we have what we call like the apoenzyme, which is just the protein portion. So it's like what we know as the enzyme. And then the coenzyme or the cofactor, right, is going to come together with that protein portion to form what we call a holoenzyme. And so the holoenzyme is the whole thing, right? If that can help you remember it. Holoenzyme, the whole thing. The enzyme plus the cofactor or coenzyme, right? So we need all these pieces to come together for some enzymes to function. Again, not every single enzyme does this, but a lot of them do, right? A lot of them have some sort of cofactor or coenzyme to help regulate the job, right, of the enzyme, the enzyme's ability to work. We also have end product inhibition. So this is usually we look at a metabolic pathway where we start with a reactant A, and we end in this case with product F, and we go through this process, right, of building, right, A, B, C, D, E, and F, right? We have all these steps with all these different enzymes to get us to an end product. But what can happen sometimes is the final product, in this case, F, right? It is accumulating in a way that we have enough. We don't need any more. And so that final product can actually go back and inhibit an earlier point in the chain. So it serves like the final product kind of serves as an off signal, right? And that we get enough of it, it binds to one of the enzymes in the earlier part of the chain, and that turns that enzyme off, right? We call this allosteric inhibition because there is a product that is binding to an enzyme that changes the shape so that we've changed the active site. We basically have eliminated the ability for a substrate to fit in the active site. This is going to basically keep us from getting too much of a final product, right? We don't want it to accumulate to the point where we can't use it all, or we've wasted supplies, we've wasted energy. And so our final product can basically turn off the earlier sections of the chain. So I mentioned allosteric inhibition and like that the final product can serve as an allosteric inhibitor. But what does that actually mean? What is an allosteric inhibitor doing to an enzyme to stop it? So here we have our enzyme and we have the active site that the substrate works with. We also have this other one called the allosteric site. The allosteric site is where the inhibitor can bind. And if we look, pay attention to this active site up here. When the allosteric site is empty, we have the nice open active site. It's usable. Everything's great. But when our inhibitor binds, look what happened to the active site. It's pinched itself off. It's changed shape in such a way that our substrate can no longer fit. So it's kind of like another way to turn something off, right? And then when the inhibitor, leaves, then we can turn back on again. Right? So it's just another way to turn things on and off. Now, what happens when you have an error of metabolism? This usually we're looking at some sort of genetic mutation, some sort of congenital disorder where a gene that codes for an enzyme is affected, right? That there is a mutation in the genetic code for that enzyme, and so the enzyme is different. It's non-functional, right? It's defective. And so because the enzyme is defective, we don't get the products that the enzyme makes, right? Because the enzyme can't work, right? The machinery is non-functional, and so there's nothing we can do to go from reactant to product in that particular pathway. There's a lot of different diseases that can occur because of loss of a product or accumulation of something. So basically what can happen, especially when we look at these more complicated chains of reactions, right? We look at these metabolic pathways. This can be because we're missing something now. There's a product that we need that we do not have in the amount that we need. We can also have the accumulation of something. So let's say like the enzyme is used to break something down from something that's more dangerous to something that's more benign. We could have the accumulation of that dangerous component, and we never get to break it down into a safer form. So we could have something like that, right? So, or maybe we make something totally different, right? There is the product that we get is not the same, and that can be harmful, right? So there's a lot of different ways that this can happen. Again, the enzyme does not make the products we need. The enzyme does not break things down that we need to break down, and we get an accumulation of something. Or it functions, but in a way that creates different products that can be harmful. So there's a couple ways we can go with this. So let's take a look at some examples of enzymatic, diseases, I guess you could say, some diseases that are caused by a deficiency or a defective enzyme, and then look at what's the abnormality and what clinical result do we see. So let's look at some that have to do with amino acids. So one is phenylketonuria, or PKU, right, for short. This is where we cannot utilize or break down a particular amino acid, phenylalanine, and so we get this increase in phenylpyruvic acid. And so this inability to basically metabolize the phenylalanine leads to intellectual disabilities, epilepsy, and whatnot. Luckily, this is something that is scanned for early on, usually a day or so after birth. We check for this now. And if we follow certain, we can follow certain protocols to protect someone from problems from PKU. Albinism, this is where we do not produce melanin, and so the person with albinism would have no melanin whatsoever, so they would be extremely pale, right? Their eyes would not have color to them either. In general, not having melanin wouldn't be a problem, except for the fact that melanin is protective against UV light. And so someone with albinism would be extremely susceptible to skin cancer compared to the average person. And so this person would need to be heavily invested in using things like sunscreens and other protective measures. It can also lead to some other issues, including with vision. Maple syrup disease, this is an increase in leucine, isoleucine, and valine. This will degenerate the brain and cause early death. Homocystinuria, this is an accumulation of homocysteine. Intellectual disabilities and eye problems can develop from this. And then some other ones, at least one that's not so sad sounding, would be lactose intolerance. Lactose intolerance is due to you not having lactase or the correct amount of lactase to break down lactose. So we can't actually use the lactose, but the bacteria in your gut do, and they produce substances that can cause things like cramping and diarrhea and flatulence. Lactose intolerance can range from being just uncomfortable to being very extremely problematic. Glucose 6-phosphatase deficiency. This is where we accumulate glycogen in the liver. We don't have the ability necessarily to break it down. We don't have the ability to stop that accumulation. And so someone here, their liver would be enlarged because we're physically accumulating something there. But we would also be hypoglycemic because we would be pulling glucose from the blood to build that glycogen. And we wouldn't have the ability to get that glycogen broken down into glucose to release it back into the blood. Glycogen phosphorylase deficiency, again, this is another accumulation of glycogen but in the muscles, and so this would cause muscle fatigue and pain. With the lipids, we have Gaucher's disease. This is going to be a lipid accumulation. So the liver, the spleen enlarge, and the brain will degenerate. Tay-Sachs disease is another lipid accumulation disease. It's a different lipid, but this causes brain degeneration, and usually death occurs by age 5. Tay-Sachs, again, this is basically there's certain enzymes that the lysosomes would normally use to break down different lipids, but they can't break it down, so there's this accumulation that develops. It starts to cause neuron death, and regularly, it is progressive. And so someone with Tay-Sachs, generally speaking, would not make it to school age. Hypercholesterolemia. Hypercholesterolemia is a disorder with a particular, with your ability to kind of break down the cholesterol in your blood. So you see cholesterol developing in like high levels. You have, so it's so high that you actually start to see deposits of cholesterol in like the joints or under the skin. It doesn't just deposit under the skin though, it also would be found in the arteries as well. So this would be someone who would be pretty much guaranteed to develop atherosclerosis in their arteries and their coronary arteries. So something that could lead to something like a heart attack. So these are just like an example. I'm not going to ask you to memorize any of these specific ones. Just understand like there is a link between a metabolic defect, a disease, and what the reactions or the clinical results we get from that.